From the macrostructures of tissue and bone down to the microscopic architecture of individual cells, human biology operates with immense complexity. Yet, if we travel deep within the cellular nucleus, that complexity gives way to a highly organized vault. Every anatomical function, every physical trait, and nearly every human disease is directed by a surprisingly simple molecular code stored in this space. The master architects of this system are nucleic acids, specifically deoxyribonucleic acid, or DNA, and ribonucleic acid, RNA. To grasp the mechanisms of human health and the targets of modern medicine, we have to examine the chemical bricks that build these genetic structures. Nucleic acids are massive polymers, but they are built from a single repeating structural unit, the nucleotide. Nucleotides are built from 3 pillars: a 5-carbon sugar core linked to a phosphate group and a nitrogenous base. The sugar determines the nucleic acid. RNA uses ribose. DNA uses deoxyribose, which lacks 1 oxygen atom on its 2nd carbon ring. A quick point of chemical nomenclature. If a structure contains only the sugar and base, lacking the phosphate group, it is classified as a nucleoside. That single missing oxygen atom in the DNA sugar backbone carries immense biological weight. It makes the molecules significantly less reactive and highly stable, perfectly suiting DNA for the long-term storage of genetic information. While the sugar and phosphate form the uniform backbone of the molecule, the 3rd pillar, the nitrogenous base, provides the variable data. These bases act as the specific letters of our genetic alphabet. This diagram displays the 2 main base families. At the top are purines, adenine and guanine, characterized by a larger double-ring structure. The bottom half shows pyrimidines, cytosine, thymine, and uracil, which are defined by a smaller single-ring framework. The distribution of these pyrimidines is strictly divided. Thymine exists exclusively within the storage vault of DNA, while uracil is utilized solely by RNA. Together, these 5 chemical ring structures provide the complete vocabulary required to spell out the biological instructions for all living organisms. Looking at this space-filling model of the DNA double helix, we see the ultimate long-term storage vault. 2 anti-parallel strands of nucleotides twist together, exposing distinct major and minor grooves along the exterior. The internal logic of this twisting ladder was brought to light by biochemist Erwin Chargaff. He observed a strict consistency in the ratios of the chemical letters locked inside. This structural diagram illustrates Chargaff's first rule of complementary base pairing. Notice the top rungs. Adenine pairs perfectly with thymine, secured by exactly 2 hydrogen bonds. Further down, we see the 2nd rule. Cytosine pairs exclusively with guanine, locking together via 3 hydrogen bonds. Because a bulky double-ring purine is always matched with a narrower single-ring pyrimidine, the DNA ladder maintains a perfectly uniform width from end to end. As seen in this replication sequence, uniform pairing is functional. When a cell divides, DNA unzips down the middle. Because A only bonds with T and C with G, each separated half acts as a precise template to build a new identical strand. The strict biochemical rules of hydrogen bonding ensure that genetic data is preserved and transmitted with exact accuracy across cellular generations. Storing the data is only half the equation. The process of turning that static code into physical biology follows a one-way flow of information known as the central dogma, from DNA to RNA to the assembly of proteins. It starts with transcription. A targeted segment of the DNA vault is unzipped, and its sequence is copied into a mobile, single-stranded format. This initiates the RNA workforce. The newly extruded messenger RNA, or mRNA, acts as the blueprint, carrying the genetic instructions safely out of the nucleus. Next comes translation. Ribosomal RNA forms the physical factory floor of the cell, while transfer RNA acts as the delivery fleet, carrying specific amino acids to the ribosome to build the precise protein encoded by the messenger. If DNA is the master blueprint locked away for safekeeping, RNA is the active fleet of messengers and builders that physically construct the organism. But this immense biochemical machinery is not infallible. We must carefully consider what happens when Chargaff's fundamental rules fail, or a replication error slips through the proofreading process. A single mutated nucleotide, just one incorrect base pairing out of billions, will transcribe into faulty mRNA, which can ultimately assemble a malformed, non-functional protein. The scale of that microscopic chemical error amplifies quickly. These nucleotide misprints are the mechanical drivers behind cancer proliferation, inherited genetic disorders, and viral pathogenesis. For clinicians and pharmacists, the reality is clear. The root cause of our most devastating diseases is often just a typographical error in the sequence of nucleic acids. Because we understand the specific chemical rules governing this code, we have the capacity to intervene. Targeted gene therapies and personalized medicine address the root of a disease by targeting the source code directly, moving beyond the mere management of symptoms. The development of mRNA vaccines takes this a step further. By delivering engineered, single-stranded instructions, we can utilize a patient's own ribosomal factories to mount an immune defense against novel pathogens. The specific chemical alphabet of adenine, cytosine, guanine, and thymine serves as the ultimate programming language of human life. Mastery of this molecular blueprint provides the definitive mechanism for modern medicine to save lives.